Hello, everyone, and welcome to this edition of the GOA-ON webinar series. My name is Michael Aquafreda, and I'm a postdoctoral researcher at the NOAA Ocean Acidification Program, and I'll be moderating today's session. This webinar series has 4 sponsoring organizations. First, GOA-ON, the Global Ocean Acidification Observing Network. Second, NOAA, the National Oceanic and Atmospheric Administration. Third, the IAEA OAICC, the International Atomic Energy Agency, Ocean Acidification International Coordination Center. And finally, last but not least, the IOC UNESCO, the Intergovernmental Oceanographic Commission of the United Nations, educational, scientific, and cultural organization. For those of you who are new to GOA-ON, it is a collaborative international network designed to detect and understand the drivers of ocean acidification and the resulting impacts on marine environments. GOA-ON serves as a platform for acquiring and exchanging data and knowledge. GOA-ON also provides information to communities, industries, governments, and global organizations who are seeking to develop action plans, best practices, and mitigation and adaptation strategies to address ocean acidification. GOA-ON has over 860 members from 105 countries. We are delighted by today's turnout, and we welcome anyone who is currently not a member of GOA-ON to join our community. During the presentation, all participants are in listen-only mode. You are welcome to type any questions into the questions box, which can be found at the bottom of the control panel on the right-hand side of your screen. We do ask that you specify the speaker you're directing your question to. We'll be monitoring incoming questions and we'll pose them to our speakers at the question and answer session, which will follow the presentations. We encourage you to pose questions and share your insights on the Ocean Acidification Information Exchange as well. And we would encourage you to use Twitter too by using the hashtag GOANWS. Today's webinar is being recorded and will be available through the GOA-ON YouTube channel. At this time, I'd like to introduce today's speakers. First, Dr. Melissa Chierici. She is an observational chemical oceanographer with more than 25 years of experience. She mainly studies the marine carbon cycle, air-sea CO2 fluxes, and ocean acidification in the subpolar and polar oceans. She is particularly interested in natural and anthropogenic drivers that are controlling ocean shifting carbonate chemistry. And she leads observational programs for ocean acidification in the Norwegian and Arctic waters since 2017. Her research also encompasses OA observations off the west coast of Africa and the Indian Ocean, which are performed on the research vessel Dr. Fridtjof Nansen. Dr. Chierici is also engaged in teaching as an adjunct professor at the University Center on Svalbard and aboard the RV Nansen. Our second speaker today is going to be Dr. Mohammed Idrissi, who is an observational oceanographer with more than 10 years of experience. He works on topics ranging from biogeochemistry to sediment dynamics to oceanography, and he also leads observational programs for ocean acidification in waters off Northwest Africa, which are performed on the research vessel Dr. Fridtjof Nansen with Dr. Chierici. Last but not least, we will be hearing from Dr. Chibo Chikwililwa, who is a marine microbiologist at the University of Namibia. She works mostly on harmful algal blooms and microalgae and has a focus on phytotoxic species and chemical pollutants in seaweed. Her ocean acidification work is primarily based around biological experiments. So without further ado, let's first hear from Melissa. So why don't you turn on your camera or unmute yourself, and I'll pull up your presentation.
Yes, okay. Thank you very much for that nice introduction, Michael. And as Michael mentioned, I'm more on the polar scientist, but today I will use my specialty as a carbonate chemist or marine chemist and doing observations in a completely different area, which I was pulled in to help out with in 2017. So it's about the science theme 10 about climate change and biogeochemical processes. And today it's focused on ocean acidification work observations that we are doing within the EIF Nansen Program, which I will tell you more about. So next slide, please. Next slide, please. Okay, thank you. Yes, so just to give you some background on this Nansen program, and I think many of you are familiar with this program, but it started in 1975, so it's been going on. So it has been using 3 different ships. All of them are called Dr. Fridtjof Nansen, or DFN in short. And their focus has been to use sustainable, the ecosystem approach to fisheries, and to improve food and nutrition security for people in partner countries. And it's NORAD, which is the Norwegian Agency for Development Cooperation. They are the owner of these ships and the program. And then Institute of Marine Research, where I am senior scientist. We are also then contributing with technical and scientific expertise. And then we have the Food and Agriculture Organization are also part of the management here. So these picture just shows the different phases, and today in the last phase with the new ship in 2017, phase 5, they have an increased focus on science implementation and also science capacity training. So and that is when I started to be involved in this fantastic Nansen program. Next slide, please. So I was asked also to add on ocean acidification observations on the ship, which was not used before, and it was also a request from many of the local partner countries that we should add these observations. And then I also said that then we need alkalinity and pH, for example. So, and then we were also working out different projects within this science theme 10. Where the main focus is to investigate the chemical environment. So opposed to the physical oceanography, we're looking at the chemical oceanography and also the change of the chemistry of the ocean. So we focus on looking at oxygen concentrations and also the extent and changes in oxygen minimum zones, nutrient concentrations and changes, as well as then ocean acidification, which has gotten a large attention in the last years. Mainly because these parameters are completely new for this program. And oxygen and nutrient concentrations have been measured since the start of the program. Next slide, please. Yes, so these are the main study areas. We are looking into the oxygen minimum zones, since with low oxygen comes naturally low pH or high CO2. And that is also when you have this upwelling of high CO2 subsurface waters coming up to the surface. And these are also processes which we can see in the Arctic and in other oceans as well. So we want to see how that will affect the coastlines, and I think Chibo will talk more about that. We're also trying to focus a little bit on vulnerable benthic habitats, so cold water corals, sponges, gorgonians, and also mollusks. So in some of the focused Nansen cruises, we also add on to look more processed and focus on these benthic habitats. And we're also very interested to look at the coastal, where we have some coastal or river runoff. So freshwater influence, because we have seen in other areas where you have a large drawdown in pH, you get the enhancement of ocean acidification in a way from freshwater influence. Everything is the relevance. The focus is, of course, the marine ecosystem and the relevance for fisheries and biological systems. Okay, next, please. Yeah, so this shows a map on the cruises. We started 2017. And the blue dots shows where we have data. So this ship goes more or less 300 cruise days every year before the corona. So it's a huge program. The ship goes several cruises and focus in some years on the west coast of Africa and some other years on the Indian Ocean. So today we will focus more on the West Africa data set, and where we are mainly in some areas we are actually collecting some of the first observations of alkalinity and pH, and then we can calculate the full ocean acidification system. And together with oxygen, nutrients, and physical oceanography measurements, we also want to explain some of the main drivers to explain the variability and distribution we see in the ocean acidification parameters. So the yellow rings shows the areas which Mohammed and Chibo will talk more about, where we have a little bit of a focused effort. And we have a good data coverage in these regions as well, on these in these areas, on these parameters. And then in 2020, we had also in the northwest of Africa, but we had to cancel the full program after Corona came. Okay, next slide, please. Yes, and this shows the 2018 data collection in the Indian Ocean, where I myself participated also on a cruise. Focus here has been also to repeat some of the historical data lines in the Bay of Bengal and some other transects from World Ocean Circulation Experiment and the Go ship cruises and things like that. So we actually get repeated transects, and from that we hope to identify trends in the ocean acidification state, whether there has been a change from the 90s to this time now, for example. We also perform regional process studies, as you can see on these blue dots. For example, around Sri Lanka, we have some fantastic and amazing high resolution data around the whole island, and it was a huge variability around that. So trying to also to explain the natural variability in these areas. Next slide, please. Yeah, so what we did within the science theme, we then added spectrophotometric determination of pH with M+/purpur indicator on the ship, together with determination of total alkalinity. And then we use open cell titration with a metron system. So these are, in a way, because we are also, one big part of this is also to have the capacity training, and then we need to do have setups that are not the big winter systems, because there will be a lot of people with different skills, and then we were settling on these systems that are a little bit more simpler to also to teach, but still gives really good high quality data. And of course we use certified reference material for this data set as well. In addition, people that are involved and joined the CTD team lab, they are also measuring dissolved oxygen with Winkler titration and salinity, and they're also involved in both collecting and filtrating for chlorophyll A and measuring chlorophyll A. And they sample for nutrients, which are analyzed usually in Norway at the Institute of Marine Research. And next slide, please. Yes, so this has been, of course, when we started in 2017, it was a huge, just to get the instrumentation on board and to get that work with very little time before the ship left Norway, and then going to Africa and being away for many years before, at least we thought so before Corona. Now it's back in Norway. But anyway, so we have had several training workshops focusing more on these alkalinity and pH measurements, as well as establishing protocols and standard routines how to do this. And they are in a way in a constant change, because you discover that you need to simplify, or you need to be more clear in your protocols, and to explain these different analyses to get everything right. So we learn a lot from that. We also have training workshops where we teach what to do with the data and how to calculate the full, how to assess analytical quality, how to calculate the full carbonate system variables using the CO2 system Excel version. So there are, it's a lot of effort, because we really want to have good data to do science of the good data. So good data gives good science. So it's a huge and major task, and also very important and lots of fun to have this training workshops. And also all the cruises, we have people from the local partners countries coming on board, and they do an excellent job to get this good data. We also have IMR team leaders, CTD team leaders, which are teachers in a way to try to establish a good protocols and also get the good data. They spend about 90% of the time teaching when they are on board the ship. We have also started to do supervision and mentoring of PhD and master students, and I think that Chibo and Mohammed will talk more about those students and what they are doing. And I'm very happy for that, because that's what we need. Fresh young people working on science. Next slide. I think that might have been the last one. Yes, so thank you very much for listening. And I wanted to make this short because I wanted Mohammed and Chibo to give them a lot of time, and I just want to introduce a little bit on the program that we are working on. And I'm really happy that you are listening and hope to meet you again. Thank you very much. Bye.
Thank you so much, Melissa. All right, so at this time, we're going to move over to Dr. Mohammed Idrissi's presentation. So, Mohammed, you could go ahead and unmute yourself and take it away.
Thanks, Mike, for the introduction and for this GOA-ON webinar series initiative. So the title of my talk will be the new marine observation of carbonate chemistry, variability, and ocean acidification state in Northwest Africa water. And this work has been sponsored by EF Nansen Program, Institute of Marine Research, Norway, and African partners such as National Institute of Fisheries Research, Morocco, Institut Mauritanien de Recherche Océanographique de Pêche, Institut Sénégalais de Recherche Agricole, et International Atomic Energy Agency, of course, which give us a lot of help. So before, next slide, please. So before I start, I'm going to present an overview of Team 10 of the EF Nansen program. I will be faster since my colleague Dr. Melissa did already, like a couple minutes ago, which this research project are under, then some result of the Northwest Atlantic coast. Next slide, please. So the science plan team of the EF Nansen program have three main research areas: sustainable fisheries, oil gas pollution impact, and the last one, climate change impacts. And the Team 10 is under the main research area of climate change impact. Next slide, please. So in February 2018, more than 60 scientists from 10 countries focusing on ecosystems of Atlantic coast Africa. And next slide, please. At the end, for the Team 10, we came up with total of 6 projects. So part of this presentation is under the project 1 and 2, and both these projects, they relay and repeat stations, sections, or historical data plus physical oceanography. And these projects are funded by EF Nansen program under Team 10, of course, and I think all African countries, especially institutes from partner countries, are involved. So please, if anyone wants to get involved, just contact me or Professor Melissa or Professor Chibo. And those are the leader of the both project is Dr. Melissa from AMR Norway. Next slide, please. I'm gonna first, Dr. Melissa, she already showed us this, but after this meeting, we found out more scientists need training and improvement protocols for good data and good science, as Dr. Melissa mentioned. So this is why we did start this kind of training. Next slide, please. So for the Northwest Africa, or the Canary Current marine ecosystem, CCLME region, which supply very significant fish resources based largely or on small pelagic fish, this fishery market contribute to economy of these countries border the sea and provide an important food and employment of coastal communities. Since 2017, the 30 years long Nansen Program, FAO and Norway began this kind of study on ocean acidification along the CCLME region. Next slide, please. So this study and the whole oceanographic cruises were carried out on board of the research vessel Dr. Fridtjof Nansen between Cape Spartel, Morocco, and South Senegal, 12 degrees. From the first one in 2017 was from between May and June 2017, and the second was from September to December 2019. Unfortunately, as Melissa mentioned too, we were going to finish the 2020 survey from Morocco to Senegal, but due to the COVID-19 pandemic, we stopped DF Nansen surveys in the south of Morocco in March 15, 2020. Next slide, please. Next. So here we will show the first result of ocean acidification state as a baseline from this new research program. So far what we did 2 complete survey from Morocco to Senegal from the 35 some degrees till 12 degrees. And during the surveys, we focused on water parameters such as ocean acidification, pH, alkalinity, total alkalinity, dissolved oxygen, salinity, nutrient, chlorophyll A, temperature, salinity, and dissolved oxygen. Again, we get the parameters also from the CTD. And the survey were performed at around 30 sections perpendicular from the coast, with a total of 110 stations in the full water column. And this include the long transit, which are just south of the Canary Island. We call it the mesopelagic transit. Next slide, please. So this showed this diagrammatic schema of the fixed station sampling regime, showing the 3 separate stations sampled along each transit at 30 meter, 100 meter, and 500 meter isobat, and 2 dry CTD stations at the 75 meter and the 150 meter isobat. Next slide, please. So for the material and method, so for each station, the parameter of temperature, density, dissolved oxygen were carried out spontaneous from surface to bottom of the water column with the CTD sound. For the total alkalinity was analyzed using the potentiometric titration. For the pH, we used the spectrophotometric pH measurement. Also, we did in order to check the functioning of the salinity and the dissolved oxygen sensors, we did analysis for both these parameters, salinity and oxygen, just for some station, just for validation. We did also chlorophyll A, seawater as a collected, the seawater collected from the CTD at the standard depth, not below, of course, 200 meters, for the chlorophyll A, and also for the few pigment measurements. For the nutrient, the macronutrient were analyzed using the autoanalyzer according to the Casablanca protocol. The other parameters describing the carbonate chemistry and ocean acidification state were derived from the total alkalinity and pH using the C2CIS calculation program and the constant Nansen, which from Dixon 1987. Next slide, please. So for the result, because of the lack of the time, I'm going to present just some result for the surface parameters for the whole area. So the value of the parameters for the temperature is low, of between 14 and 20, and it's located north of Bouche d'Or, and its high value height were observed from the south of Bouche d'Or to Senegal for both seasons, 17 and 19. For the salinity, salinity remained relatively constant near 36.5 through the surveys of 17 and 19. However, small patches of salinity values approaching 34 and sometimes 35 and sometimes 34 was observed, particularly in the southern region of the whole area. Next slide, please. The concentration of the oxygen, the solved oxygen in 2017 is higher than 2019. Generally, in 2017, it's around 400 micromole per kilogram, and in 2019 are below than 300 micromole per kilogram. And I apologize for the color. It's supposed to be green for the fluorescence and chlorophyll A, so we put another color. I don't know why. So the fluorescence shows a higher value, and near shore with it's over 3 milligram per meter cube, and it's homogeneous in water masses for the rest of the area. Next slide, please. So unfortunately for 2017, we have some gaps for both pH and total alkalinity, just for 2017. So the value of the pH are between 7.9 and 8.1 and 17, and some area has values below than 7.5. In 2019, the values are between also 7.9 and 8, and we observed the increase of the pH over 8 in the southern area, especially South Mauritania, Senegal, and Gambia. For the alkalinity, the total alkalinity almost still have the same value, around 400 micromole per kilogram. However, also small patches of total alkalinity value less than 2200 observed, particularly in 2017. Next slide, please. For the nutrient, the value of the nitrate are almost the same, and they are around 5 to 10 micromole. The value of the nitrate are around 0.2 also, and 17, the values of the nitrate are lower than 90, especially in the coastal zone and area in north of the whole Northwest Africa, especially in Morocco. Next slide, please. For the phosphate, the value for the phosphate and silicate value in 2017 are higher than 19, and we still working on the data, all data, of course, to combine between them. Next slide, please. So what we found so far? So far, we found large variability along the coast connected to salinity changes, primary production, temperature and biology processes. In fact, the measurement parameter pH to alkalinity coincide perfectly with the physical parameters, temperature and salinity, while the calculated parameters inversely varies. In this project, we are working to show the first ocean acidification data from this area, water, and discuss the variability in context of deep water intrusion, salinity changes, and temperature and phytoplankton production. Next slide, please. So we are going to move forward to the Science Plan 21-22, what we are going to do and what we have done already. So for the data 17 and 19, so we have the data, raw data, post-processing, quality check. We're done with this all of it. For the data 2020, we still have the raw data, and we will complete this data 2020 with the next surveys, of course. And we are looking to do sampling for 21 using Dr. Fridtjof Nansen or Moroccan vessels from, and if it's Moroccan vessels, it will be from Cap Spartel to Cap Blanc, from Tangier to Mauritania. And we still have to combine the data from 17, 19, 20, and 21 if we get it for all the data set, just to study this kind of variability and describe major processes in different regime of the Northwest Africa water. We still put in PhD students since December 19 in this program. And so far, we put for this project, what I am presenting just like part of it, a PhD student on which she's working in this part. But it's just gonna be Morocco, so it's gonna be from Cap Spartel to Cap Blanc, and will be based for the whole time series from 17 to 21, or we are going to do 22 with this PhD. And also we did put some master degrees for this program for just couple regional part, especially for Morocco. And also we are looking to put other kind of master or PhD in other partner countries. And here I'm talking about Mauritania, Senegal, or Gambia. And thank you so much.
Thank you so much, Mohammed, for that. And at this point, I will introduce Dr. Chibo Chikwililwa. Chibo, do you want to turn on your camera for a moment and unmute yourself? And while you're doing that, I will pull up your presentation. Okay, Chibo, you can take it away.
Good morning. Good morning, good afternoon, everyone. You need to go back to the beginning. Okay, my name is Chibo Chikwililwa. Mike, you tried, well done. So I'm going to be essentially talking about the Benguela Current itself, because OA work in Namibia is still in its infancy. So I'm going to give you an overview on the area itself, as well as how OA aspects fit into the Benguela and why it's of importance, as well as what is actually happening with respect to the Nansen. And as you can see with my hands flying, I'm going to switch off my camera so I can focus. Next slide. So as most of you know, the Benguela is one of the eastern boundary upwelling systems. This is a wind-driven system. And what it has in connection with the Canary upwelling system, which Mohammed was talking about, is the fact that we have seasonal upwelling, nutrient-rich water, even though it's low in dissolved oxygen and low in pH. And the important part about this is the fact that because the water is nutrient rich, there's a lot of biological production. As when the water upwells, it comes into the euphotic zone, you'll be able to get to a lot of primary production with respect to the autotrophs such as phytoplankton. Now, the reason why these E-buses are so important is because even though they encounter a small surface of the ocean, they still tend to have quite a large amount of global primary production itself. And if you look at it from that perspective in relation to ocean acidification, the way in which the link falls in is the fact that this primary productivity takes up the PCO2. Next slide. And the way in which it does this is essentially using the biological pump, which is the schematic diagram I have on the side, where you're taking dissolved inorganic carbon, it's taken up by the phytoplankton, and then that essentially makes its way down the water column. In a situation where there was nothing else in the water itself, it would just go down to the floor. But of course, that doesn't happen. Now, the reason, even though there are two aspects that affect the way in which the marine carbon is cycling, the biological pump is the one that's the most important for E-buses. And it is with respect to that you actually can find that they're not all inherently sources or sinks. They can either be one or the other or both, or depending on the season that you actually find them in. Next slide. So the way in which the Benguela upwelling system fits into this is the fact that it has high primary productivity. In relation to the E-bus, it actually accounts for 40% of the primary production. This whole range is truncated between the Benguela frontal zone on the north and the, Agulhas Current in the south. So these are two subtropical zones that actually house the area itself. And it's also subdivided into the northern and southern Benguela about Lüderitz, which is in that circly yellow bit that you can see over there. So because we say it has high primary productivity rates, then it stands to reason that the biological pump would be very strong. And for coastal regions, it's dominated by diatoms and dinoflagellates. Next slide. Whereas if you move offshore and you have the more mature upwelled water, the phytoplankton community that dominates in that area are nanoflagellates, the plankton community, or nanoflagellates and coccolithophores. So the point at which you can ask the question of why the northern Benguela would be such an important aspect with respect to the carbon cycle is due to the fact that in comparison to the southern Benguela, the northern Benguela is actually seen as a carbon dioxide source. Next slide. So this is due to the fact that the northern Benguela has a high rate of remineralization. So the particular organic matter that's actually made from photosynthesis is actually broken down quite readily. So a study in this area found that the factors that actually affect how the DIC is taken up on all those aspects depends on the strength and duration of the upwelling, because that actually has an effect on the nutrient load as well as how the phytoplankton would actually produce with respect to that. And then the second aspect is also the extent of the aerobic decomposition of the organic matter that stems from this. Now, unfortunately, the data that was based on seeing this area as a source was from offshore samples. And offshore samples tend to only have nanoplankton as well as coccolithophores. So there could be a very large discrepancy with respect to that from that. So we would need some way in which to actually incorporate the level of coastal variability that actually comes predominantly from this biological pump. Next slide. So the reason why the NBUS is so important with respect to this, if you look at that figure in the corner, that brown water is actually from the Walvis Bay Harbor, where they're growing oysters and other shellfish such as mussels. That water is so brown and thick with phytoplankton that when we put a Secchi disc down, it actually disappeared within the first 10 centimeters. So this biological pump in this coastal area is really, really of great importance. So the fact that we have this undersampling and the fact that we have some influence with respect to the upwelling means that there are so many parameters in there that we need to have an understanding of so that we can get an idea of how this area actually affects not only the regional carbon cycle, but also the carbon cycle globally as a whole. Next slide. So even though the carbonate chemistry / OA parameters are an area that we have not really looked at in this area, one of the other things that is of importance here, which has a very long data set from the Ministry of Fisheries, is the fact that we actually have oxygen minimum zones along the coastal areas. So you'd find that the bottom water in some instances never really goes above 0.5 milliliters per liter of DO. And the data from this, the reports from this have realized that there's some level of seasonality with respect to that. And also you tend to have a wider range of hypoxia during warm stratified conditions of summer and autumn. Now, these conditions also can lead to sulfidic water in the water column, and this would stem from anaerobic decomposition of organic matter. So there are other drivers that are in play in this region that also have to be factored in when you're looking at the OA parameters themselves. And next slide. And that sulfidic water that came by, essentially, I'm sure most of you have seen this, if you think of this area where you get hydrogen sulfide eruptions. So what you find there in that image on on my right-hand side is elemental sulfur that you could actually detect using satellites. And this only occurs during specific periods of time where you have to have a combination of methane bubbling, a water column that's very low in oxygen, as well as particular organic matter that's broken down by the anaerobic microorganisms. And then you'd have a leak of hydrogen sulfide, which is normally used by a sulfide oxidizing bacteria. And I just put a picture there of Thia margarita because, to my knowledge, it's still one of the largest microbes in the world. So the northern Benguela has all of these features, but the bit that's lagging behind is looking at ocean acidification, because if we look at climate change, these are other parameters we have to have an idea of. Next slide. So what that culminated into was what Melissa and Mohammed were talking about, in the sense that in 2017, we had the first Nansen cruise, where this was actually the first instance where pH and total alkalinity were actually recorded. And pH specifically, spectrophotometrically, because there have been a few instances where they've been using the MBS scale, but of course, I mean, you can't really use that. And for the 2017 and 2019 data, those aspects were done. So here I'm essentially just showing you the raw data that hasn't been processed yet. Next slide. What this project enabled was that we were able to get the data from, if you look on my right-hand side, it's along the southern coast of Angola, right through to the northern sector of Namibia. So each of those, okay, I was on this cruise itself where essentially in between driving around with a boat looking for pelagic fish, we would essentially do a transect as we went along. So you can see from the temperature data where you'd actually go, okay, the way that it looks is like each transect is essentially a dot on each of those of those figures, where you have your depth going from surface down to the bottom of the water on your vertical axis, and the horizontal axis is the time. So we essentially went from north to south. So the temperature shows you a movement from the subtropical waters in Angola with high temperatures into the more subtropical, into the more temperate waters of Namibia itself. And the oxygen data itself, essentially, if you look at the top bit, that probably is from, what do you call it, photosynthesis. And an area to highlight is the red spotches in the corner there, which are probably from high bloom masses that you would find in Namibia, because that's the period of time that went into Namibia. And then the pH itself has no trend, because again, I say this was raw data and it still has to be processed. So in order to overcome these problems, next slide. We were very lucky that NORAD was able to fund a student. So she's a master's student, her name's Cecilia, and she's actually going to be working under the theme 10, which is the one that Melissa and Mohammed introduced. And what's great about this is that even though her overall aim is to look at the impacts of climate change on DO, nutrients, and ocean acidification in the northern Benguela, one of the first things she has to do is actually collate all of that information. So what she'll be doing is essentially the first steps in telling a story of the let's say the OA status in this region, and there can be a comparison between 2017 and 2019. So we're really, really excited that she's there. And then this is one of the other aspects where, because the Nansen project goes all through the Western Africa region, this is where we could actually get a collaboration between the Canary and the Benguela upwelling systems. Because essentially, if you think about it, this is a vessel that's going through all the other areas. And the advantage is that we're using the same equipment, the same quality assurance, so we get good quality data, and that data can actually in some way be compared with respect to the different regions itself. So that's one of the areas that we can feed into. That's why Cecilia's role is so important, because she will be the one that will set up that data and set up that information. Next slide. The reason why I put this in here is because one of the important things that came out of this Nansen project, wasn't only the fact that this was the first time we're actually measuring, OA parameters. We're able to take that capacity because they trained a lot of staff members from the Ministry of Fisheries as well as from the university. So we're able to take the skills and knowledge that we learned from that and feed it into this regional project, because more and more people are starting to understand that climate change and OA go hand in hand, and we need to have an idea of what is actually happening in order to see if we can actually mitigate some of these aspects. And it's a regional project, so it includes Angola, Namibia, and South Africa. And I speak under correction, but I think also for Angola, 2017, that was the first time they'd actually gotten, the high resolution data of the high resolution carbonate chemistry data for the pH and total alkalinity. Next slide. So essentially, this project is going to go a step forward in the sense that, yes, we will conduct more monitoring so that we can also get some aspect of seasonality that doesn't necessarily have to be linked to the Nansen cruises. So we can get some seasonality aspects to it. And of course, which is also very important, which was great about the training that we got on the Nansen, accurate carbon chemistry data. So this project will be able to fund that. And also of importance is the fact that we're going to actually start biological experiments that can actually use these aspects and parameters that we actually learned how to use as well. And the idea of this is to get an idea of the variability, as well as looking at how key species in each region are affected by OA. Next slide. So I will end off here because this is essentially just a schematic of a schematic aspect of what I was talking about, in the sense that we're setting up the basic monitoring, essentially just adding pH and TA since all the other parameters were being done as it is. And it'll be along the transects that you can see on my right-hand side of the screen, as well as focusing the biological experiments on micro and macroalgae. And with that, you can go to the next slide. I'd like to say thank you for listening.
Thank you so much, Chibo. So I'm going to pull up my presentation at this point. And I could ask all of our speakers to turn on their cameras, and I'll check the questions box to see if we received any questions for the audience members. If you have any questions, please type them into the questions box. And I also encourage you to call out which speaker you're directing your question to. Sorry, it takes a moment to get the questions pulled up. Okay, so the first question we have is for Melissa. And the question is, are there trainings for data collection and processing available online for new students? I think particularly since COVID has made traveling such a challenge, I know a lot of institutions are moving into the virtual realm. Is that something that you're looking into?
Yes, we have discussed it, and it also started before Corona, because I couldn't go to one of the training workshops in South Africa, and then I had a webinar or some lesson on the web to discuss, to show some. So we have been thinking about it also more and more now with the Corona situation. Some challenge we have is that sometimes, as you say, the bandwidth hasn't been very good. So that is something to work on. But definitely that is, and also the people that are on board and helping out in the CTD lab, they get, of course, that's the best training you can get if you participate in one of the cruises. Then you will also get the package for how to estimate the whole protocols, and also how to do things with your data afterwards, data processing, you would get also with you. But that's the package, teaching package we have, you get also when you do the training workshops. But perhaps that we have to develop that better, yes.
And speaking of data, another question came in asking what the policy was for the data that comes from the DFN cruise. Is this data that's going to be publicly accessible? Where could someone access this data if it is publicly available?
Yeah, it's a little bit different. It's a little bit different than like a normal research cruise, since there are so many partners that are involved, and we also have. So we go through contact points in the different partner countries, and then we discuss what to do with the data. And we also need to protect PhD students and master students that are using those data. But eventually, the aim is to have them publicly available.
Hopefully
it will probably take a while. Yeah.
Yeah, and hopefully one day on the GOA-ON data portal.
Oh yeah, yeah.
All right. So the next question here is for Chibo. The question is, do you collect plankton samples for taxonomy? And do you assess biodiversity? And if so, or if not, are such data stored in a standardized, interoperable format that can be shared on databases like OBIS?
Because I do not collect the data myself, because I am more focused on the coast and I collect samples and then I basically culture them. I can put you in contact with a person at the Ministry of Fisheries that has that data, but it's not accessible to anyone. Maybe if you ask him nicely. His name is Dion Lowe and he works for the Ministry of Fisheries. So he actually has that data because they have, they essentially do, what do you call it? Monitor. They have a monitoring program. So they have a lot of data. And yes, it's, I think at the moment, it even goes down to the nanoflagellates, I think. But it does have the different classes, so the dinoflagellates and diatoms. But I speak under correction.
Well, that's a good important segue to note that all these questions, we actually received a lot of questions, and I don't think we'll have time to get to all of them, but I will be posting all of these questions on the Ocean Acidification Information Exchange. So maybe there, Chibo, you and Frank, who asked this question, could connect, and you could provide him that name of that person at the Ministry of Fisheries. So I think our last question is going to be for Mohammed. And the question is, what has the response of the Canary Current upwelling system been to the warming? And do you have any ideas how that will affect its productivity? Can you hear me, Mohammed?
Tough question.
Yeah.
Yes. Can you repeat again the question, if I can catch it?
The question is, what do you suspect the response of the Canary Current will be to the warming that you observed?
Oh, from what we just from the DF Nansen program, from the cruises, what we have so far from 17 to now, to till 2020, I don't think so. We can say something about it. So we cannot relate it what's going on so far from the CCLME. But if I'm going to back from what we have, for example, for Morocco, it will be something different. What's going on exactly in our area? If the impact of the CCLME current, the current CCLME current, of course we have a big impact of what's going on and for the warmer water. I don't know if I...
Yeah, it sounds like what you're saying is you don't quite have enough data yet from the cruises to pull out some of that. Melissa, do you want to chime in?
Yes, I also want just to complement that you have to think about that the ship has 300 cruise days per year. So we collect about more than 3,000 data points for alkalinity pH every year in a normal year, pre-corona. So one good thing with corona for this program has been that we have actually had time to start really looking into the data. So it's not really a lack of data now. I mean, of course, you can always get more, and you want more, for example, these mesopelagic lines, as Mohammed showed, going further offshore. That's what we also want to do, and do it on repeatedly in a long, long time. But so we have been started to really investigate and work scientifically on the data. And that's when you also discover the quality of the data and things like that. So that has been a good thing with the corona.
Mm-hmm. Excellent. Well, I want to thank our presenters one last time. Thank you so much for joining us today and for sharing this really exciting work. I want to thank our audience for joining us today. We had a really excellent turnout, so thank you. And before we leave, I also want to announce the next webinar in the GO-ON webinar series. It'll take place on Wednesday, July 14th at 10 a.m. Hobart, Australia time. The presenter is Dr. Eric Mortensen, and he will be presenting his talk, which is entitled Regional Changes in Southern Ocean Biogeochemistry Due to Projected Carbon Uptake. And you can register for that webinar at the GO-ON website. Finally, if you are interested in presenting a talk for the GO-ON webinar series, we're always looking for more speakers. Go over to the GO-ON website and you can fill out a registration form and we'll contact you. Thank you all very much, and we'll see you in a couple weeks. Bye, everyone.
Thank you very much. Bye-bye.
Thank you.
Thank you.